Good day. Today I'm going to talk about HIV prevention using monoclonal antibodies, or as we call them, broad neutralizing antibodies. So let me first put this into context. We use integrated strategies to prevent HIV. We'd like to make a vaccine, but we don't have one. We have other biological tools that help us. Of course, the mainstay of HIV prevention is treatment as prevention. That's on the far right of this slide. In the yellow circle is the idea that we can use antivirals and now broad neutralizing antibodies, we hope, prevent the acquisition of HIV. And so this is really kind of ushering in the era of pre-exposure prophylaxis or PrEP. And that era was ushered in with the use of the pill tenofovir FTC, Truvada, approved many years ago, 8 years ago, as a once-a-day way to offset the acquisition of HIV. The strength of this is that the idea really works. The weakness is that you've got to reliably take the pills. And so there's been a lot of interest in longer acting strategies to prevent HIV acquisition. And the long acting strategies might include injectable formulations as opposed to, or maybe longer acting pills, or maybe rings that could be left in for a long period of time, antimicrobial rings, antiviral rings. If we look at what's available, we know Merck is got a drug called islatravir that's a once-a-month pill. We know Merck and other companies are working on implants. We know that there are needles and patches. We know that there are sub-Q capsid inhibitors, and we know that the long-acting injectable agent integrase inhibitor cabotegravir is very likely to be approved for both treatment of HIV and for the prevention of HIV. In recent studies completed, cabotegravir demonstrated very reliable prevention of HIV in both men and women when given every 2 months by injection. But I'm now going to turn my attention to antibodies. And this slide emphasizes the kind of natural biology of HIV infection. There's the viremia. After an eclipse phase, there's viremia, and antibodies are generated, which are used to demonstrate seroconversion. But those antibodies do not really help the host fight off the infection. Now, some hosts, maybe as many as 50%, make strain-specific neutralizing antibodies against whatever they're seeing at that moment in time. The problem with the strain-specific neutralizing antibodies are that the virus replication lacks fidelity. So as it evolves, whatever antibodies are generated are no longer effective. We'll come back to this idea later. Untreated HIV infection can lead as many as 20% of people with infection to form broad neutralizing antibodies. These antibodies will kill lots of different strains of HIV, but they won't protect the host or help the host fight off the infection. But these broad neutralizing antibodies have attracted a lot of attention, are really the subject of our conversation this morning. And in this slide, and this whole kind of field really goes back about 10 to 20 years, maybe as long as 20 years, with Doug Richmond and Dennis Burden and others leading the way. And then about 10 or 12 years ago, the field really took off. And Nicole Doria-Rose published this paper in which she was able to demonstrate, I think it might be animated, she was able to demonstrate that about a quarter of donors make weak neutralizing antibody response, but about 25% in this particular collection of volunteers generated broad neutralizing antibodies, more potent neutralizing antibodies. Now, in this slide drawn by Bart Haynes and his group, it kind of emphasizes how this happens, that the virus causes the infection and the host generates an antibody. And the virus evolves to avoid these antibodies. And in the untreated host with replication ongoing, there's an endless arms race where antibodies are chasing different viruses. And eventually, a B cell has undergone enough mutations or somatic hypermutations such that it's making one of these broad neutralizing antibodies. This idea of studying sequentially the virus and the antibody generation has become a popular theme in many labs because it's important to understand the potential for vaccine development, where vaccines might lead to a broad neutralizing antibody. Now, so as labs got more and more interested in broad neutralizing antibodies, their targets became critically important. And on this slide, kind of colorful slide, you see the HIV virus and its insertion into the cell membrane, and you see different sites where antibodies have been, where broad neutralizing antibodies have been isolated and generated for the purposes of scientific study, and as well as for the purpose of making a drug. And you'll see that the CD4 binding site is really quite attractive and critical. And most of the antibodies that have been tested are directed at the CD4 binding site, some at the mPER site, some at the anti-CD4 site. And it really isn't a brand new idea. As shown on this slide, as early as 1990, investigators had the idea of taking polyclonal antibodies and trying to protect non-human primates from infection. And they did that successfully with the right cocktail of antibodies. And then in 2000, we had Dennis Burden's group and others working on the first monoclonal antibodies to protect animals. The challenge studies got better and better. And then the current generation of monoclonal antibodies were used successfully to protect non-human primates. And through their successful protection of non-human primates, they really generated a lot of interest in the idea of antibodies as treatment or adjunctive to treatment, and antibodies as an alternative for pre-exposure prophylaxis. And in the universe of these monoclonal antibodies, one antibody made by the NIH Vaccine Research Center, VRC01, became the source of a variety of important studies. Now, here I have to have a kind of way a disclaimer. I helped to run one of the leaders of the HIV Prevention Trials Network, which is partners with the HIV Vaccine Trials Network. We are NIH supported. We are then partners with the Vaccine Research Center. So a lot of our work focuses on products generated through the NIH Vaccine Research Center. And I would emphasize, while I'm going to focus on the work we've done in this space, there are other investigators working on monoclonal antibodies in different universities and in different organizations all over the world that are doing excellent work with other monoclonal antibodies or other broad neutralizing antibodies. So my focus is not meant to be exclusionary. It's really meant to be an example. So the VRC01 antibody, the first issue was to take it from its ability to protect non-human primates from infection and then move on to the idea that you could use it to protect humans from infection. And in order to try to pursue this goal, the drug was put into phase 1 studies. And on this slide, you see doses of 50 micrograms and 10 micrograms and the pharmacology of these and how long the drug might last. And in the initial phase 1 studies, the drug lasted a fairly long period of time, suggesting you might be able to give it every 8 weeks, depending on how much concentration was required to neutralize strains of HIV. And you can get a sense of the potency when you gave the doses shown on this slide. Now, VRC01, then we get to the idea of, well, how do you know that this antibody is sufficiently broad to kill lots of strains of HIV? And there's a kind of, there is a laboratory configuration of testing a panel of viruses that are lined up in tiers that would allow claims that an antibody has sufficient potency against a wide number of strains to be tested further. So VRC01 became the focus of our attention. And after demonstrating its safety and reasonable potency, it was concluded that we needed to do a proof of concept study. Now, I want to emphasize proof of concept. It was never believed that the antibody was going to be sufficiently broad or durable to become a drug. And we'll get back to that in a little while. But we did believe that we could test whether the idea was correct, whether the antibody had the potential to neutralize HIV during the process of acquisition in at-risk hosts. And so we set up the antibody-mediated prevention studies worldwide. And as I said, these are proof of concept studies. One was for men who have sex with men and transgender women, and the other was for women. And the questions we were asking were pretty straightforward. Could we give this drug safely with minimal reactivity and no serious adverse events? And to what extent could we prevent HIV infection over 80 weeks in subjects at high risk for HIV? So these subjects had to be negative in enrollment, and we were trying to prevent infection at some rate. And then co-primary or secondary goals of the study are to understand how much VRC01 antibody would be required for neutralization in vivo, not in a test tube, in vivo. And then when breakthrough isolates occurred, would we be able to understand how these breakthroughs occurred? And of course, in the best of worlds, the breakthroughs would be resistant to the antibody, just a few of them, and occurring at times late after antibody delivery, when one would predict that the antibody concentration might have waned below the concentration required for neutralization. And so the study was set up, as I said, worldwide, to compare a 30 milligram per kilogram dose of VRC01, a 10 milligram per kilogram dose versus a placebo in between 4,200 and 4,600 subjects, each getting 10 infusions, leading to a study that would last 80 weeks with an additional follow-up to 104 weeks. Study was done ultimately in 47 sites in 11 countries, and we infused probably at this point more than 50,000 doses of VRC01 at the concentrations I've shown you. One of the most surprising things of the study was how loyal the subjects were. That is, once they started on the study and were coming in every 8 weeks, there was very little loss to follow up. There were no serious adverse events, as I've indicated. And the study subjects just were retained at a level that was quite remarkable, given that they had to come in and get an infusion every 8 weeks. And this really affected our thinking about feasibility of long-acting agents. Now, we completed the AMP studies. And what I'd like to do, of course, is to tell you all the results today. But I really can't do that because we haven't told the study subjects all the results, nor have we presented this at a meeting, nor have we published the results. So I want to give you kind of a broad insight without telling you the results. So the broad insight is on the same slide I showed you a few minutes ago. Well, first of all, is it safe to infuse VRC01? Yes, it proved very safe, as I've already indicated. Second, we were trying to prevent infection as best we could over an 80-week period. And we have the results of our ability to do this, and they'll be presented, as I said, in meetings in January and in manuscript form, so the results are forthcoming. Could we understand the concentration of VRC01 in vivo required for neutralization? And what are the limits of a single antibody? And as I'll tell you, we can answer both those questions with the data we generated. And then for breakthrough isolates, we are able to study them. When I say we, there are hundreds of scientists and dozens of labs working on this study. And these scientists worldwide are able to identify really critical information, understanding the nature of infection or the acquisition of the virus in the face of the antibody, and identify lots of information about breakthroughs. So as I've already indicated, the trial is completed. The results will be made public in early 2021. And maybe by the time this talk is shown, the results will have been made public. So I apologize for what would seem to be secrecy, but this is kind of where we are at this moment in time. Now, just a brief diversion, because what happened with COVID, which has dominated all of our lives, is HIV investigators were repurposed to be COVID investigators. And so the HVTN, the HIV Vaccine Trials Network, and the HPTN, the Prevention Trials Network, fused together to form the COVID Prevention Network, which is asked to work on vaccines that you've heard so much about and monoclonal antibodies you might have heard about as well. Now, I would emphasize that it is the work of Dennis Burden and Scheid and Nusenzweig and many others whose technologies kind of allowed monoclonal antibodies then to enter the COVID space. And this very colorful slide just shows the basic idea. The spike protein has to bind to an ACE2 receptor, and antibodies should be able to block this binding process. All the vaccines generated, all the vaccines tested are being tested to generate such antibodies in high concentration, but passive infusion of a monoclonal antibody would offer maybe 1,000-fold more antibody at a moment in time than a vaccine could offer. And there is no shortage of interest then in monoclonal antibodies for COVID, informed and inspired by the HIV field. This is a list of maybe 50 monoclonals at different companies that are in different stages of development. But the train has really left the station, and Eli Lilly and Regeneron have completed studies of using monoclonals for treatment, and both companies have received emergency authorization for these monoclonal antibodies as for early symptomatic patients. And then in collaboration with the NIH, Lilly and Regeneron are working with the COVID Prevention Network on using the same monoclonal antibodies or monoclonal antibody cocktails for the prevention of acquisition of COVID, either as pre or post-exposure prophylaxis. So the kind of PrEP field that the HIV field developed exists in the COVID field. Now, back to HIV. What's next? Well, first, we'll report the results of our AMP study, but the AMP study was always a proof of concept. And it was always a consideration that we would need monoclonal antibodies or broad neutralizing antibodies with more potency, at least 10 times more potency, with the broadest possible coverage and with longer half-lives, longer than 2 months, and perhaps through sub-Q injection. So this is just an example of the VRC01 patient who is, I believe, died many years ago, but before treatment was available. The same blood samples over time could be used to find an even more potent antibody, VRC07, which could have a mutation in the FC receptor, leading to a longer half-life. So VRC07523 is more potent and lasts longer. N6 was from a different donor, but has a similar binding site and is even more potent and can be modified to last longer. And N6 is, I think the rights to N6 are owned by GSK. And what happens is, as shown on the slide, we get into the idea that in order to use monoclonal or broad neutralizing antibodies for pre-exposure prophylaxis, we're going to need to combine antibodies. And this shouldn't be a shock. We always anticipated this. The heat map shows the micrograms per mil IC80, and you see when you get to greater than 50, you can see holes in different antibodies. You see VRC07 is pretty good, but it has some holes. And PG121, PDGM 1400, pretty good, but have some holes. And then on the right, you see when we combine these 3 antibodies together, we get our very best cocktail if we're going to combine 3 antibodies. Now, this same logic can be applied in a kind of a homemade synthetic antibody. So Sanofi has made tri-specific IgG, in which they've taken PDGM 1400, 10E884, and VRC01, and made a synthetic antibody, which, if I show you the heat map, you can see the tri-specific Sanofi antibody, which if it's tolerated in early phase trials, becomes a really potent drug that saves us the trouble of mixing 3 broad neutralizing antibodies together to get the very best cocktail. And so, and of course, COVID slowed everything down. All the work we were doing was kind of not ground to a halt, but really slowed down by COVID. But Sanofi has been very committed to developing this product, and the NIH has been very committed to the tri-specific, the combination of antibodies that I showed on the preceding slide. Now, I want to be clear, as I've already indicated, the antibodies that we're talking about that are owned or that were generated both by the NIH and by other partners in the NIH, they're not the only games in town. Other investigators have made many, many really important broad neutralizing antibodies, and I would draw particular attention to Michelle Nuzensweig's group, where they use a cocktail of antibodies that have been very capable of protecting animals from infection, and have been working in collaboration with the Gates Foundation in development of this potent cocktail to be used sub-Q to attempt to prevent HIV infection in clinical trials. And that's not the only lab group that is doing this kind of work. So I want to be dispassionate about how many people are working on this problem. So I guess the issue as I come to the close of this is, this has been an unusual couple of years. We have and if you look at the last 10 years, it's remarkable how far we got. We have completed the proof of concept AMP studies, and we think the results will inform the field. We are preparing now, getting ready to try and make combinations of antibodies or using synthetic antibodies as PrEP that would last for many months as an alternative to antiretroviral agents. And we have then been forced to deal with COVID at the same time. And I think that this slide from New York Times emphasizes a point. The biggest monster, COVID's a monster, but it's not the only monster. And our need to deal with tuberculosis and HIV and malaria, which was the point of this article, are no less than they were before COVID. So I would put our work in context and hope that in the next year or two, we can get back to working full-time on monoclonal antibodies, that is broad neutralizing antibodies, for the effective prevention of HIV acquisition. Let me point out that the slides I've shown, I cannot take credit for the most part. My colleagues, Dr. Kaup, Mascola, Corey, and others, and especially Mascola, I liberated slides from them in order to give the talk I've just given, and I really appreciate their generosity with their slides. Let me point out that the studies that led to all this work involve, thousands, literally thousands and thousands of study subjects, hundreds and hundreds of investigators, and then there are more than 100 laboratories and scientists working now worldwide on the specimens we generated in the AMP studies. So I think you could anticipate we'll publish or report a lot of results from the work that has been completed. I also want to especially thank the funders. We've had tremendous support from NIH across several institutes, and they're listed at the bottom of this slide. So thank you for listening, and I look forward to answering questions in the live session. Thanks.